Today we will discuss how the digested nutrients are absorbed in the gastrointestinal tract. So in the intestinal lumen, the large macromolecules which are present in food are digested into simple substances which are now been converted into the absorbable form. So they have to cross the intestinal epithelium and reach the blood vessels, and from there through blood they will be carried to different parts of the body. And this intestinal epithelium, it is made up of cells. So the substances or the end products of digestion are to be transported through the cell membrane of these intestinal epithelial cells. And we know that the cell membrane is made up of a lipid bilayer in which there are proteins which are interspersed in between. And suppose this region, it is the lumen of the intestine and this is the cell interior, and the digested nutrients have to traverse this membrane to reach the cell interior from this intestinal lumen. So this is the place where digestion has taken place and the end products of digestion are here, and from there the substances have to come into the cell. So they have to cross this barrier, that is the cell membrane, which is made up of lipids and proteins interspersed in between. And these substances can either directly pass through the lipid bilayer or they have to pass through the proteins. They have to pass through the channels or tunnels which are created by these proteins. And there are two mechanisms that exist for the transport of substances. One is diffusion and the other is active transport. Diffusion can also be called as passive transport. We will first go to the basic difference between this passive transport or diffusion and active transport. In diffusion, a substance will move from a region of its high concentration to that of its low concentration, that is along the concentration gradient. Whereas in active transport, the substances are made to move against the concentration gradient, that is from a region of low concentration to a region of high concentration. So for diffusion, there is no extra source of energy required. It is by the kinetic energy that the molecules will travel from one place to the other. Whereas in active transport, apart from this kinetic motion of matter, there requires an additional source of energy because the molecules are to be transported against the concentration gradient. So we can say diffusion is an energy independent process, whereas active transport is an energy dependent process. And another difference is, for active transport, there is always the requirement of a molecule which is called as carrier. So active transport always requires the need of a carrier, which is usually a protein molecule which is present in the lipid bilayer of the cell membrane. Whereas there are two types of diffusion. One is called simple diffusion and the other is called facilitated diffusion. In simple diffusion, the substance can directly pass through the lipid bilayer of the cell membrane, or it can pass through channels which are there inside the proteins present in the lipid bilayer. So the substance will move either directly through the lipid bilayer or through channels which are already built inside the channel proteins. In facilitated diffusion, the molecule will not be able to pass through directly. It has to take the help of a molecule which is called as a carrier or a permease, and that is also a protein which is there integrated with the cell membrane. Permease molecule which binds to the substance to be transported and undergoes some conformational change so that the substance is transported to the other side of the membrane. But the similarity which they share, that is simple diffusion and facilitated diffusion, share a similarity that they both are energy independent because the substance is transported along the concentration gradient, that is from a region of high concentration to a region of low concentration. Whereas in active transport, energy is required, and energy is usually derived from the hydrolysis of ATP molecule. Active transport is carrier dependent and ATP dependent, and it is against the concentration gradient. Simple diffusion is energy independent and carrier independent, and it is along the concentration gradient. Facilitated diffusion is energy independent, but it is carrier mediated, and it is also along the concentration gradient. And the rate of simple diffusion, it is determined by certain factors. That means how much of substance is available for diffusion. What is the velocity of the kinetic motion of these substances? What are the, how many pores and openings will be there in the membrane to allow the transport of these substances? So, the surface area of the membrane, that is also a factor that determines the speed of simple diffusion. Now, in facilitated diffusion, the substance to be transported will chemically interact with the carrier molecule or the permease molecule, and this permease will shuttle that particular molecule through the membrane in this chemically bound form. Simple diffusion is energy independent, it is carrier independent, but still this is the slowest process to occur, because once the concentration on both sides become equal, the process will stop. And similar is the case with facilitated diffusion also, but it is faster than simple diffusion because there is involvement of a carrier. Active transport is considered to be the fastest. We can see facilitated diffusion. Here, this is the substance to be transported. This is the carrier protein or the permease which facilitates the transport of the substance, and there occurs a chemical bonding between this substance and the binding point of the carrier, and now binding will lead to a conformational change in the carrier protein, and the substance can reach the other side of the membrane. So, this is how facilitated diffusion occurs. Now, we go to active transport. It is the uphill transport that takes place against a concentration gradient. It is again divided into two types. They are primary active transport and secondary active transport. The difference between primary active transport and secondary active transport lies in the source of energy used to cause the mechanism. So, we will first go to primary active transport through an example. So, here we can see a pump. This is a protein which is integrated as part of this membrane, and it acts like a pump. This is called as the sodium potassium pump. The sodium potassium pump works in such a way that it will forcibly pump 3 sodium ions out of the cell to the cell exterior, and it will pump 2 K plus ions or 2 potassium ions from the cell exterior to the cytoplasm. So, it will pump sodium ions outward through the cell membrane, and it pumps 2 potassium ions from the outside to the inside. So, this pump now creates a sodium potassium concentration difference along the cell membrane, and thereby a net negative charge is created on the inner side of the membrane and a net positive charge is created on the outer side of the membrane, since it is pumping 2 K plus ions in for every 3 Na plus ions pumped out, and so there will be a net positive charge on the outer side of the membrane and a net negative charge in the inner side of the membrane. This is very essential in the transmission of nerve impulses through neurons. This mechanism is it is playing key role in nerve impulse transmission. So, here this pump is the carrier or the permease which is transporting sodium and potassium ions, and for deriving energy for this particular process, it is having an enzymatic activity. It is having a portion that has got an enzymatic activity, that has got ATPase activity. What is ATPase activity? It is able to break down ATP into ADP and inorganic phosphate, thereby deriving energy. And it is this energy that is being used for pumping sodium ions outward and potassium ions inward. So here the energy is directly derived by this particular protein, which will split ATP into ADP. That is why it is called as primary active transport. So in a nutshell, in primary active transport, the energy is derived directly from the breakdown of ATP or some other high energy phosphate compound by the carrier molecule itself. What is secondary active transport? This is also called as co-transport. So, as it was already mentioned, the sodium potassium pump can maintain or it can create an electrochemical gradient of sodium and potassium. There will be higher concentration of sodium outside the cell, which is created by the activity of the sodium potassium pump. So, a concentration gradient exists now. That is, there is higher concentration of sodium outside and there is its lower concentration inside. So this gradient represents a storehouse of energy because the excess sodium which has been pushed out will have a tendency to diffuse into the interior. It will always have a tendency to move from high concentration to a region of low concentration. So this gradient which has been created by the sodium potassium pump gives an urge or compulsion for sodium to move again back into the cell interior. If conditions are proper, this sodium ions which will start diffusing into the cell interior can also pull other substances along with it. So the diffusion energy of sodium will pull other substances along with sodium through the cell membrane. So here the transport of two substances are coupled with. One will be sodium and the other will be a partner molecule, and so it is called as co-transport. So this is one form of secondary active transport. So for sodium to pull another substance along with it, it cannot itself come into the cell carrying the other substance with it, but it has to resort to the help of a carrier. So another carrier protein is being used. We already met with one protein, that was the sodium potassium pump, which all created this electrochemical gradient. It has pushed sodium out, so sodium had a tendency to come in, but sodium cannot now resort to the sodium potassium pump to come in. It has to go to another carrier protein. So this is the second carrier protein to which sodium comes for help, that is, it has to come into the cell, and this carrier protein also allows another substance to be co-transported with sodium. So glucose can be co-transported with sodium in this way. In that case, the new carrier protein can be called as a sodium glucose symporter. So now finally, who is transporting sodium and glucose? The symporter. But it is not the symporter which has derived energy. It has no ATPase activity. In primary active transport, it is the carrier protein itself which is having ATPase activity, but here we can see that this protein is not having any ATPase activity. Instead, it is using the diffusion energy of sodium. And why there is diffusion energy of sodium? It is because the sodium potassium pump through ATPase activity had created an electrochemical gradient of sodium or concentration gradient of sodium. So this gradient energy instead created a diffusion energy in sodium, and that is why sodium came and bound to this particular carrier protein, and this carrier is now ready to take up glucose also. So that is why it is called as sodium glucose co-transport, and once these two molecules are attached, the energy gradient of sodium will cause both these molecules to be transported into the interior of the cell. So that is why it is called as secondary active transport. because in secondary active transport, the energy is derived secondarily from an energy that has been stored or created in the form of ionic concentration difference. We can take the example of sodium. So, there was a concentration gradient created by primary transport or sodium potassium pump, and it is that energy derived from the concentration gradient that allowed the second carrier to act. That means it allowed the second carrier to transport sodium and glucose together. So, here the energy derived from primary active transport is used for secondary transport. But so these are the differences between primary and secondary active transport mechanisms, but how they are similar is that in both there requires the presence of carrier protein. In the first case, the carrier itself derives energy. In the second case, the carrier derives energy from a concentration gradient that was created by another protein, which can be a pump. And what difference is there between the carrier protein, which is involved in active transport, and facilitated diffusion? The carrier protein which takes place in active transport, say for example the sodium potassium pump, it is capable of imparting energy to this transported substance. So, in this case, it is capable of transferring energy to sodium or potassium to move against the concentration gradient, to move against the electrochemical gradient. But in facilitated diffusion, as we saw earlier, in facilitated diffusion, this is not giving any energy, or this is not deriving energy from ATP, hydrolysis or anything, and it is not imparting energy to this molecule, but it is only creating a way for this molecule to be transported to the other side of the membrane. So, here there is no ATP hydrolysis, but in active transport, this carrier, it is capable of imparting energy to sodium to move outside against the concentration gradient, potassium to move inside against the concentration gradient. So, that is the difference between the carrier proteins which are involved in active transport and facilitated diffusion. And now we will see the substances that are actively transported through some cell membranes. They include sodium ions, potassium ions, calcium, ferrous ions, hydrogen ions, chloride ions, iodides, urates, several sugars, and most of the amino acids. They are all transported through active transport. After going through the various mechanisms of nutrient absorption, we will go to a concise picture of which all places in the gastrointestinal tract are involved in absorption of nutrients. So, the total quantity of fluid, which includes the absorbable substances, amounts to about 8 to 9 liters per day. So, that means about 8 to 9 liters of fluid are to be absorbed in our gastrointestinal tract a day. So, how does this come? Out of this 8 to 9 liters, the total ingested fluid which has undergone digestion amounts to about 1.5 liters, and the rest around 7 liters is contributed by the gastrointestinal secretions, the gastric juice, the saliva, the intestinal juice, the pancreatic juice, the bile, etc. And most of these substances are to be reabsorbed. So, the total quantity of fluid that is to be absorbed each day by our intestine amounts to about 8 to 9 liters. And most of this is absorbed in the small intestine itself. Only 1.5 liters will pass into the large intestine from the small intestine, and the rest is absorbed in the small intestine. So, we have to understand that. So, small intestine is the prior place of absorption of nutrients. The stomach, whereas, is a poor absorptive area, because unlike the intestinal mucosa, the stomach lacks villi, and the epithelial cells in the stomach are joined by tight junctions which will not allow enough substances to diffuse through them. So, because of all these reasons, the stomach is showing poor absorptive activity compared to the intestine. But there are some substances which are absorbed in the stomach. For example, some lipid soluble substances which can traverse through the lipid bilayer, alcohol, aspirin, etc. They are absorbed in the stomach. And mostly those substances which will diffuse by simple diffusion through the plasma membrane will have lipid solubility. So automatically some of these substances can pass through any part of the gastrointestinal tract. So also in the stomach some lipid soluble substances will be absorbed into the epithelium of the stomach mucosa. Whereas when we come to the intestinal mucosa, we can see that the intestinal mucosa first of all shows a number of folds, valvular folds, and these valvular folds are called as folds of Kerckring or valvulae conniventes. So these are all folds which are seen on the intestinal mucosa. And in these folds there are many finger-like projections which are called as villi. So primarily there are these folds of Kerckring which in turn contain finger-like projections called as villi. And hence we can imagine how much the absorptive surface area will be increased. So this is the best possible mechanism by which surface area of absorption can be increased in the intestine, that is the presence of valvulae conniventes and the presence of villi on them. Now, when we take a cell, an epithelial cell which is lining each villus, so these are the villi, and if we take one epithelial cell from each villus, you can see that the cell membrane of the epithelial cell, it is drawn outward into a number of minute finger-like projections. These minute finger-like projections which extend as cytoplasmic extensions from the apical or upper side of the cell are called as microvilli. So they appear like the bristles of a brush, that is why it is called as brush border. So each intestinal epithelial cell which is located on a villus will have a brush border which is having around 1000 microvilli. So each cell itself will be having 1000 microvilli, and each microvillus can be of length 1 micron and diameter 0.1 micron. So they also tremendously increase the surface area of absorption. So first there will be the folds of Kerckring, on which there will be the villi, and on the villi there will be the microvilli, which will lead to very higher increase or a tremendous increase of absorptive surface area. And when we take a longitudinal section through a villus, villus is a finger-like projection. So when we take a longitudinal section through the villus, what we can see is its lumen is supplied with capillary network. So these are the blood capillaries. This is the artery that enters into the villus. This is the vein that leaves the villus, and this is the capillary network in between. Apart from this, inside the villus we can also see a central lymph vessel which is called as lacteal. This is also very important because it plays a major role in the absorption of fats. So the internal anatomy of the villus shows a highly intricate network of capillaries and a central large lymph vessel which is called as lacteal. So when this is the longitudinal section, you can see this is the transverse section of a villus where this is the centrally placed lacteal, and this represents the capillary network. So this is how the intestinal mucosa is specialized for absorption. Now we go to the absorption of substances one by one. So we take the absorption of water. Water absorption takes place by diffusion, and it follows the laws of osmosis, that is, water always flows from a hypoosmotic solution to a hyperosmotic solution. So when it is the chyme or the partly digested food from the stomach that comes to the intestine. So the chyme is there in the intestinal lumen. If the chyme is dilute, definitely water will be absorbed into the intestinal mucosa, because it is the chyme which is dilute, that is the intestinal lumen is hypoosmotic, or it is having a higher concentration of water, and so water will diffuse from the intestinal lumen into the intestinal epithelial cells, and thereafter it will be taken up by the blood vessels of the villi. So it strictly follows the laws of osmosis. Whereas in converse, water can also be transported in the opposite direction, that is from the intestinal epithelium back into the lumen or back into the place where the chyme is present. When does this occur? When the chyme is hyperosmotic, when the chyme is having lower amounts of water. That is, if the discharge from the stomach, suppose if the partly digested food coming from the stomach to the intestine is highly concentration, or if it is hyperosmotic, there is a chance for water to diffuse back from the intestinal epithelium into the lumen. So that is one method of transport of water through osmosis, that is from the lumen to the intestinal mucosa or back from the mucosa to the lumen. So that is one direction. So here you can see this is what was mentioned, that is from the intestinal lumen water is diffusing into the intestinal epithelial cells. We can also see another transport of water, another place where water transport occurs, that is from the intestinal epithelial cells, the cytoplasm of the intestinal epithelial cells, to the spaces between the cell. These are called as paracellular spaces. That is, the spaces seen between the lateral side of the cell are called as paracellular spaces. So water transport not only occurs from the lumen to the intestinal epithelium, but it also occurs between cells. That means through the paracellular spaces. So these are the two sites in which we can see water, the osmosis or diffusion of water. Now we come to the absorption of ions into the epithelium. So what are the ions absorbed? We can see the absorption of sodium, chloride, bicarbonate ions, calcium ions, iron, potassium, magnesium, phosphate and some other ions. So we will first go to the absorption of sodium ions. So sodium is absorbed through diffusion, but for diffusion of sodium from the lumen to the cell interior, that is, sodium has to diffuse from the lumen to the cell interior. In the lumen there should be a higher concentration of sodium. That is, usually in our intestine, the intestinal lumen will have a sodium concentration of 142 milliequivalent per liter, whereas inside the intestinal epithelial cell it will only be 50. Since such a gradient exists, sodium will have a tendency to diffuse into the intestinal epithelial cell cytoplasm. Now why there is such a concentration gradient? Why this is always 50? If it increases, the diffusion will slow down. If the concentration on both sides become equal, the diffusion will stop. So for maintaining this gradient, what happens is that the pumps located on these membranes, they will forcibly pump sodium out into the paracellular spaces, and that is active transport. So there is active transport of sodium taking place from the intestinal epithelial cell cytoplasm into the paracellular space, and because of this, the sodium concentration will always be 50 milliequivalent per liter, and here the concentration is 142 milliequivalent per liter, and therefore sodium will diffuse from the intestinal lumen into the cell exterior, cell interior. So the motive power for sodium absorption is provided by the active transport of sodium from intestinal epithelial cells through the basal and side walls of the cells into the paracellular spaces. and this active transport is the energy for active transport is derived through ATP hydrolysis. Then next is the transport of chloride ions. The chloride ions are negatively charged, sodium ions are positively charged. So when sodium is absorbed, chloride also follows it. That is, the negatively charged chloride ions will be passively dragged by the positive electrical charge of the sodium ions. So chloride will accompany sodium, and when sodium comes in, water will also have a tendency to assist sodium. So it is coupled with or it is associated with water transport also. So when sodium is brought in, there will be osmotic gradient created. The lumen starts becoming hyposmotic when more and more sodium comes in, and because of this, water also starts coming in. So we can see that whenever there is diffusion of sodium inside to the interior, water also accompanies. Then we go to the absorption of bicarbonates. Bicarbonates are also absorbed in the upper small intestine because a lot of bicarbonate ions are secreted through pancreatic juice, through liver as bile. So all these bicarbonate ions cannot be lost through the feces. Bicarbonate ions are secreted for the purpose that there should be neutralization of the acidic chyme, and the pancreatic and intestinal enzymes require a moderately alkaline environment, and for this purpose bicarbonate ions are being released. So they are to be reabsorbed. We cannot afford for the loss of these bicarbonate ions through feces. So they are absorbed in the small intestine. Calcium ions are actively absorbed into the blood, especially in the duodenal region, and the amount of calcium absorption is in such a way that it will meet our daily requirement of calcium. And there are two factors which are involved in the absorption of calcium. One is a hormone that is secreted by the parathyroid gland, and it is parathyroid gland which increases calcium absorption depending upon our body requirements. Another one is vitamin D. Vitamin D is also required for calcium absorption. Along with these ions, iron is also absorbed into the small intestine, and along with that, ions such as potassium, magnesium, phosphate, etc. are also actively absorbed through the intestinal mucosa depending upon our bodily needs. And when we examine, we can see that monovalent ions are absorbed easily than polyvalent or bivalent ions. So when we compare, the bivalent ions are normally absorbed in small amounts compared to the monovalent ions. When we take an example, the maximum absorption of calcium ions is only 1/50th as that of sodium ions. Sodium is monovalent, whereas calcium is bivalent. But it doesn't cause any problem to us. Because of this disparity, there is not much problem to us because our body, most of our biochemical reactions that take place in our body are more dependent upon monovalent ions. So that is why it is arranged so. So these are the various ions that are absorbed through different ways in our gastrointestinal tract.